Okay, thanks for viewing the previous video, guys, and I'll try to do a great job with the rest of these figures here as well. So for figure 3, for at least for part A, they wanted to, and for all of figure 3, they wanted to evaluate the human grafts histologically 1 month after transplantation. And you can kind of see that in the paper as well. But for part A, they looked at a ventricular wall and saw comparable scarring for all 4 groups. So for the progenitor group, the cardiomyocyte group, the mononuclear group, and the non-cardiac group, you can see this kind of blackening along the wall here. And you can see that, like even though that these 2 groups and even this group is considered as a treatment, there's still scarring on the ventricular wall 1 month after transplantation. And for part B, they kind of quantified that for us. And of course, you can see that with the treatment groups, they're a little bit smaller, but it's not really any. There's no significance in reducing that scarring or that infarct size or anything like that. Um, so they can't really conclude that these cells lead to a reduced scarring or infarct group or infarct area. For part C and D of figure 3, they wanted to look at and demonstrate that the cells have actually engrafted, and which you can kind of see in figure C and these various figures in D as well. But in figure 3C, they pointed out that all of the hearts contained islands of grafts that were encapsulated by scar tissue. And they also noticed in the article that human cells were only found in 3 of 11 recipients of the non-cardiac derivatives, which is a good thing for these figures in that the non-cardiac derivatives shouldn't really produce any viable results. The grafts were pretty small for those and didn't contain any cardiomyocytes, which was also good. But the main takeaway from these figures is that the cells that they were using actually engrafted, which is good because if they want to develop a treatment based with these cells, they need to make sure that they engraft into the injured heart. And lastly, for figure 3, they wanted to look at the graft area. So they noticed that for cardiomyocyte groups, they were a bit larger than the cardiac progenitor groups. Even though there was no significant difference, they pointed this out, and I think it's an important point to bring up because it depends on the infarct size of whether or not you need like a certain graft size, or even depending on the heart structure or geometry, that could play tremendous effects as well. And as well, in both groups, the cardiomyocytes expressed myosin heavy chain, cardiac troponin, and NKX 2.5. And those are all kind of markers that represent the heart and heart cells. So especially for cardiomyocytes, it's important for them to express these markers, because then you can actually say that, hey, the progenitor cells developed into cardiomyocytes and are actually progressing well and growing and assisting with cardiac repair. So for figure 4, the researchers wanted to look at revascularization after 1 month of cell injection. So to do this, they used immunostaining. So for part A and B, you can kind of see those results. For A, they used CD31, which we've all known is the endothelial marker. But more importantly, they used the anti-human specific method, and that kind of gave them more specificity, which is what they truly wanted in evaluating the graft as well as the revascularization. And for part B, they used RECA or RECA, which is rat endothelial cell. And again, that's for specificity purposes. But one thing that was important from both of these figures was that they saw greater capillary density in the host tissue instead of in the graft. And you can kind of see that in here and as well as here, that it's kind of the opposite of what you'd expect. You'd expect greater capillary density inside the graft because that's kind of where the cell should be located. But that kind of leads me to kind of think, based on one of the other figures I showed in the introduction video, of how blood vessels actually revascularize into the graft and not from the graft. So maybe there's some kind of signaling pathway that causes that, that maybe somebody hasn't explored yet, or that at least I haven't explored yet. So for figure 4, C, D, and E, it's kind of the quantified results from this immunostaining. But what they saw, and we'll break it down one by one. So with 4C, they were looking at capillary density within the graft between the progenitor groups and the cardiomyocytes. And from what they saw, there's not really a significant difference within the graft for these two groups. And they measured it here using a dimension, and yeah, you can see there's no significant difference. For 4D, what they wanted to look at was capillary density within the infarcted area. And for this, they used all 4 groups. And here you can also see, some changing results, but overall no significance between these groups within the infarcted area for capillary density. And then for 4E, they noticed that there's a higher capillary density in non-infarcted areas. And you can kind of see here, they've displayed their significance values and whatnot. But this is kind of truly strange because you'd expect, some kind of change within the infarct or within the graft. But instead they saw it not in the infarcted area. So that kind of still leads me to kind of believe there may be some kind of hidden mechanism, some kind of signaling maybe, that causes revascularization outside and then flowing into the graft. And it doesn't really occur within the graft, but yeah, this is kind of the opposite of what they were kind of expecting. So in the next video, we'll talk about some of the summary and conclusions from the article, as well as, discussion and experimentation and directions as well.